Hey guys, today's at FID for January 17th, 16th, January 16th, sorry. A classroom assignment will appear after you watch this video. There will be a roll sheet or an attendance sheet in the Google Classroom. Make sure you fill that out, please. Yesterday, we had been talking about meiosis and the possible outcomes of the production of gametes. Today, we're going to kind of connect that to genetics, and we're going to do so in a real simple way. We're going to look at Mendelian genetics. And Mendelian genetics is probably what you remember doing maybe from 7th grade or maybe from 10th grade or 9th grade bio. What we're going to see in Mendelian genetics is a real simple pattern of inheritance, and it makes it real easy for us to understand and gives us a real straightforward pathway to discussing genetics and inheritance patterns. We call it Mendelian genetics because of a monk, an Austrian monk named Gregor Mendel, who liked to garden. He was a hobby gardener, but he kept meticulous notes. And he noticed some really interesting stuff with his pea plants, and he kept just copious notes of the outcomes of the crosses he did. And this allowed him to make some great insights into the patterns of inheritance in pea plants. And we know many traits in other organisms follow these same patterns. So really kind of a groundbreaking discovery. And this was done at a time well before we understood genes and DNA and anything like that. So that is just kind of just like made on inference and kind of just genuine curiosity and critical thinking that Gregor Mendel possessed. So anyway, when we look at Mendelian inheritance, and the reason why we study it in bio classes is because it's real straightforward. It's real simple. Mendelian patterns of inheritance follow a simple dominant and recessive pattern. The dominant trait typically obscures the recessive trait, or we don't see the recessive trait when dominant is crossed with recessive in true breeding crosses. These genes are typically unlinked, so that stuff we were talking about yesterday relating to linkage, we don't see linkage in Mendelian patterns of inheritance. And we can represent Mendelian patterns of inheritance very straightforward in Punnett squares. And we can do real basic math calculations to get the outcomes, and that makes it real easy for us to figure this stuff out without having to draw a Punnett square. We can just do a quick calculation. So Mendelian genetics can be fun. Mendelian genetics is very easy to get the hang of. And it's a great learning tool and a great stepping stone for the more complex genetic stuff because we can see these patterns of inheritance absolutely. We don't have to like wonder. Later on, we'll talk about more complex things like incomplete penetrance and epistasis, where one gene's function depends on another. But for right now, we're just going to talk about Mendelian inheritance. So Mendel, in his crosses, in one case, crossed tall plants with short plants. He crossed plants that he referred to as true breeding tall with plants that he referred to as true breeding short. The true breeding tall plants never produced any offspring other than tall offspring. And the true breeding short plants never produced any offspring other than true breeding short offspring. And this worked out really well. So what he did was he crossed true breeding tall plants with true breeding short plants. He took and pollinated them by hand using a little paintbrush or something. He then took the offspring of that cross and allowed them to self-pollinate. Okay. In the offspring of that second cross, he observed the following outcomes. He observed that roughly 800 of the plants were tall, and roughly 200 of the plants were short. Okay, 787 and 227. Okay. And turns out this is like a 3-to-1 ratio. And he thought that was puzzling. And he did this numerous times with numerous other trait pairs. And he found similar outcomes. And this really got him thinking. And he came up with the idea that some traits were dominant and some traits were recessive. And he actually kind of figured out a little bit relating to the way this happened. And as I said before, this was pre-understanding of chromosomal inheritance. So that was kind of an interesting thing for just a curious person to come up with. So if we were to look at this today, the way we would set up our problem, we would represent the tall plant a certain way, and we would represent the short plant a certain way. Um, in the case of the tall plant, we would say that because it's true breeding, it only has one type of allele. Okay. It only has one type of allele, and that is the tall allele. So even though it can possibly pass along two different tall alleles, it really only has a tall allele. So we're going to say it's big T, capital T, as a representation of that. And um, this represents those alleles that can be passed on. And if you recall, alleles are like versions of a gene. Okay. So they are genes, but they're like versions of it. There's, in the case of this trait, there's the tall and the short allele. In other traits, maybe there are more than two copies or more than two types, but for the purpose of Mendelian discussion, we're looking at stuff that has either one or the other. There's a absolute dominant and absolute recessive. We represent our short, true breeding short plant with two lowercase t's. Those represent the recessive trait. Okay, so symbolically, that's how we're going to represent this. It's worth mentioning that while Mendel called his plants true breeding, we would now use the terminology homozygous. We know that they have the same allele. Both copies of the allele that they possess are the same. Homo, prefix homo means the same. Okay. Um, and the terminology can be used interchangeably. So just be aware of that. The term hybrid and the term heterozygous will be used interchangeably as well. Okay, so be aware of that. All right, so if we do a Punnett square, here we look at a basic Punnett square showing the Mendelian cross of a tall parent crossed with a short parent. Okay, so this is how we set up a Punnett square. We put the alleles possible from one parent on top and the alleles possible from another parent, the other parent on the side. Okay. And then we just kind of played Battleship. If you've ever played old school Battleship, remember you had to kind of like go from the side and over from the top or down from the top. And you'd say like A1 or whatever. So we're just going to drag our letter down from the top and over from the side, and we're going to fill in our squares. In our parental cross, okay, we had a true breeding tall with a true breeding short. In our parental cross, we see that all the outcomes are the same. Every offspring here is heterozygous. Everyone is heterozygous. They're all identically going to appear tall, but they're all going to carry that short allele. They're heterozygous. Okay. We refer to these as the F1 offspring, the F1s. So these individuals are referred to as the P generation, not like P plant, but letter P, parental. And the offspring of that are referred to as the F1s. Okay. So what you'll recall is Mendel did this cross, and then he allowed the F1s. He planted the Ps from the P cross. He planted them. He let those F1s grow and self-pollinate. And this is what he saw in the next round. So again, we took the F1 parent and the other F1 parent. Okay. And again, we're talking pollen here. We're not talking like they had to mate or anything like that. And these are the outcomes we got. In this box, we get a capital T, capital T. That's a homozygous dominant. In these two boxes, we got heterozygotes. And in this box, we got a homozygous recessive. Now, from a phenotypic standpoint, from the way these things look, these three, the dominant homozygote and the two heterozygotes, are all going to be tall plants. This is going to be a short plant outcome. And again, this box doesn't represent this box doesn't represent the offspring. It represents possibilities. Okay, that's a really important thing. Uh, Guys, you sometimes get hung up on the fact that there's like four outcomes here. These represent four possible outcomes. They don't represent four offspring. Okay, they could represent four offspring, but they really represent outcomes. Okay, and we refer to these as the F2. Okay, so when we look at this, we would say, oh, there's a three tall to one short ratio, 3 to 1. Okay, and that's called a phenotypic ratio. We also need to talk about the genotypic ratio. The genotypes are the specific allele combination outcomes. Here I see 1/4 is this outcome. I see 2/4 or 1/2 is this outcome. And I see 1/4 is this outcome, the homozygous recessive. Okay. Those numbers are going to be important here in a second. The question I ask here is, what if there is a greater number than just a single pair? What if we're looking at more than one pair of traits? And the answer is that happens all the time. And this is where math comes in. This is where math becomes very helpful. If I cross two plants that were heterozygous for tall, so heterozygous for tall would be big T, small t, and heterozygous for purple. Purple's dominant, so big P, small p. I represent that here. Could I do a Punnett square? Sure, this is a 16 box. Remember, you might have seen that before. We'll do one in class next time we meet. Sure, I could do that. That would be pretty easy and pretty straightforward, but on a test, I don't have time for that. Could I determine the chances of any of the outcomes just mathematically? And the answer is yes. So in that F1 cross, we saw from the previous slide, 1/4 of the offspring were homozygous dominant, 1/2 were heterozygous, and 1/4 were homozygous recessive. The flower pattern, the flower inheritance pattern is the same as the height inheritance pattern. So we can do some simple math to figure this out. Okay, so let's look at the next slide. I asked some questions here. If I had this possible outcome, big T, big T, big P, big P, what are the chances that I get that? Okay, so if I look at this and I'm going to do a little annotate here. Okay, if I look at this, big T, big T, big P, big P. What are the chances that I had big T, big T? 1 in 4. Okay, 1 in 4 times chance I got big P, 1 in 4. What's the outcome there? What are the chances I get that? And the answer is 1 out of 16. Okay, pretty straightforward, right? All right, let's look at the next one. Big T, small t, heterozygous. Okay, what were the chances that I had a heterozygous? That was 1/2. So I would take 1/2 times heterozygous again, 1/2. What's 1/2 times 1/2? You can do basic multiplication. You can do this. The chance I get that is 1/4, 1 in 4. Okay. What about the chance that I get a heterozygous for that first trait, but a homozygous recessive for the second trait? Again, that's 1/2. But in this case, it's not going to be 1/2 times 1/2. It's going to be 1/2 times 1/4 because 1/4 is the frequency that I would get this outcome in just that regular monohybrid cross. So what's 1/2 times 1/4? It's 1 in 8. Okay. And this last one, again, what if I had this outcome and that the homozygous recessive outcome with the homozygous dominant outcome? And that's going to be 1/4 times 1/4, or as we mentioned, 1 in 16. Okay, pretty straightforward stuff. I'm going to give you a few problems like this. That's going to be the assignment. You're going to have to do a couple of these little simple problems. You don't have to submit them online. You can submit them on paper. Okay, we can submit them on paper next time we meet. I'm going to give you about 8 problems. Okay, maybe a couple just basic Punnett square problems too. Anyway, guys, please make sure you stay safe today, stay warm, and I will see you soon.